Bioelectricity, aka the electrical activities that biological entities such as humans or animals emanate, has long been understood as a consequence of ion movement across biological membranes governed by ion channels, pumps, for example, as well as transporters responding to voltage as well as chemical gradients. Now, this view has been extraordinarily successful in explaining, for example, firing within the neuron of a brain or muscle contraction as well as cardiac rhythm in our hearts. However, it has also encouraged a reductionist tendency to treat ion channels as largely independent molecular devices whose collective behavior emerges simply from their kinetics as well as distribution. This review article that we will be discussing today, the structural electrical biology or the architecture of the bioelectrical code, challenges this assumption by arguing that bioelectric signaling is fundamentally shaped by spatial organization at multiple scales, from nanometer level membrane separations to tissue level architectures. In doing so, this paper proposes that bioelectricity should be understood as an emergent property of biological structure rather than as a purely molecular phenomena. The authors of these papers situate their argument within the context of recent advances in structural biology, especially in cryogenic electron microscopy and cryogenic electron tomography. These techniques have made it possible to visualize these ion channels, scaffolding proteins, for example, and membrane interfaces with unprecedented resolution in near native states, which is extremely impressive. With atomic structures of channels have been available for some time now. The ability to observe, for example, how these proteins are arranged within intact cells and tissues has revealed an additional layer of organization that cannot be inferred from isolated structure alone. This paper then argues that this ultra structural context is essential for understanding how bioelectrical signals are generated, constrained, and propagated in living systems. A central theme of this article is that the importance of ion channel clustering and the scaffolding proteins that organize these clusters. Ion channels, for example, rarely function as uniformly distributed elements across a membrane. Instead, they are concentrated into specialized domains whose size, density, as well as the molecular composition are tightly regulated. Scaffolding proteins, on the other hand, anchor channels to cytoskeletal elements, align them with signaling patterns, and define the distances between opposing membrane. These distances are not arbitrary. Small changes in intermembrane spacing can dramatically alter local electrical field, ionic concentration, and current flows. The authors then argue that such nanoscale geometric effectively encode electrical behavior, forming what they describe as bioelectrical code within the biological entity. This notion of a bioelectrical code does not imply a symbolic or genetic code in the traditional sense per se. Rather, it refers to a set of physical constraint and spatial relationship that determine how electrical signals arise and interact with biological tissues. From this perspective, voltage, for example, is not merely a variable to be measured, but a consequence of structural design. Membrane, for example, or channel arrangement, extracellular cleft width, and protein organization collectively shape the electrical landscape in which cells operate. This review also suggests that ignoring these features lead to an incomplete understanding of bioelectrical phenomena, especially in complex tissue such as the brain and the heart. Okay, so one of the strengths of this paper lies in its treatment of ephaptic conduction, which has historically been underappreciated due to the difficulty of studying it experimentally. Ephaptic effects require extremely tight control of extracellular space, for example, often on the order of tens of nanometers. Such spaces are invisible to conventional light microscopy, for example, and they're very difficult to persevere with traditional electron microscopy techniques. And the authors here then argue that modern cryogenic methods are uniquely suited to resolving these structures and may reveal that ephaptic interactions are more widespread and functionally important than previously assumed. This reframing has implications for how neural synchrony, cardiac arrhythmia, as well as collective electrical behaviors are understood within biological entities. Now, it's important to note that this article does not present new experimental data per se. Its contribution is conceptual and integrate rather than empirical. It synthesizes findings from multiple fields, which is extremely important, and reframes them through the lens of spatial organization. This makes this paper more interpretive than definitive, if you will. But it's not speculative per se because it uses a lot of existing scientific research. Their arguments are grounded in established, for example, physical principles and supported by existing structural and physiological evidence, where the authors are very careful, though, to frame their conclusion as a call for further investigation rather than as a final theory per se. Now, why should we care? The potential implication of this particular perspective extends beyond our basic science. If, for example, bioelectric behavior is strongly shaped by structural organization, then pathological changes in tissue architecture could have direct electrical consequences even in the absence of molecular defects. This could help explain why some diseases are difficult to attribute to single gene mutations, for example. It also suggests new therapeutic strategies aimed at restoring or modifying structural relationship rather than targeting individual ion channels. At a broader level, this paper gestures towards deeper questions about the development, evolution, as well as cognition. Bioelectrical signals play known roles in, for example, embryonic patterning and regeneration, processes that depend critically on spatial information. So, understanding how electrical signals are encoded by structure could shed light on how complex biological forms arise and maintain themselves. While the author stopped short of making strong claims about, for example, consciousness, they suggest that any serious account of higher order biological function must take bioelectric architecture extremely seriously. In other words, the structural electrobiology argues that bioelectricity emerges from the spatial organization of biological matter as much as from molecular dynamics. By highlighting the role of ultrastructure in shaping electrical behavior, this paper challenges reductionist models and calls for a more integrated approach to electrophysiology. It is a scientific review with a clear conceptual stance, aiming to redirect attention toward the architectures that make bioelectrical signals possible. While many of its ideas remain to be tested experimentally, the framework per se proposes is very grounded. It's also very timely and likely to influence how bioelectric phenomena are studied in the coming years. And this perspective offered by structural electrobiology aligns very closely with the principles of symbiotic intelligence, especially the idea that intelligence and regulation emerge from continuous interaction between structure, signal, and environment rather than from isolated computational units. In biological systems, for example, bioelectrical behavior is shaped by spatial arrangement of the membranes, channels, as well as the extracellular spaces, which together constrain and guide the signal flow of electricity. This mirrors a core premise of symbiotic intelligence as discussed in this channel, namely that meaningful information processing arises from coupling between sensing, internal state, and physical embodiment of intelligence within biological entities. Just as ion channels require architectural context to produce coherent bioelectrical signals, intelligent systems require tightly integrated interfaces between biological signals and artificial substances to achieve adaptive and context aware behavior. From this standpoint, symbiotic intelligence can be viewed as an extension of the bioelectrical code of biological entities, such as humans, into a hybrid biological slash technological system, aka the next generation of AI, such as AGI, or the next generations of humanoid. In a sense, biosignals such as heart rate, brain waves, and so forth are not merely just data streams to be decoded algorithmically, but they are expressions of an underlying physiological intelligence architecture that are shaped by evolution of millions of years and are constrained by the physical structure of the biological entities. And so, through the integration of these signals into the next generation of artificial signals, we can then imagine a future where the biological intelligence is seamlessly and beautifully and intelligently integrated within the next generations of humanoid or AGI or what have you. And so that is the one of the main cores of symbiotic intelligence discussed in this channel. I'll add the link to this fascinating paper below. Feel free to click on it and read it. And thanks so much for all your comments and shares and feedback. I really appreciate it. See you guys next time. Bye.